recording. This is the second part of Chapter 2, and we'll continue talking about chemicals. Now, we introduced the idea of polarity among molecules, that one side of a molecule can be very positive and another side can be very negative. Now, water is this way because the hydrogen end of the water does not have a very strong affinity or pull on its electrons, and so the hydrogens become somewhat positive, whereas the oxygen has a much greater affinity for the electrons. The electrons hang out, for the most part, on the side of the oxygen, so you have a negative and a positive side. Now, assuming that this person is married to this person, is it conceivable that he might also be attracted to this person? Well, we're going to assume he is an oxygen, and these are both hydrogens. He's bonded to this one, but because of his negativity, he is attracted to her positivity, though being bonded with this positivity. The same thing happens with water molecules. You see, the positive aspect of the water molecule is attracted to the negative aspect of the adjacent water molecule. And so water sticks together. And this bug is walking on water because of surface tension created by those hydrogen bonds. And what happens in his case is that the force of gravity pulling down on him is not as strong as the force of the hydrogen bonds pushing up on him. So we call that surface tension. So water is attracted to itself, which is a major player in the chemistry of life. Okay, chemical reactions occur when chemical bonds are formed, rearranged, or broken. And we represent them as a chemical equation, which has a molecular formula for the reactant and the product. So here we have the reactants, and this should be an arrow, but it didn't translate very well. Two hydrogens become hydrogen gas, which are the products. Or 4 hydrogens and a carbon become methane. Now, synthesis reactions, and again, our arrow did not translate from one program to the next. A plus B goes to AB. So synthesis is always bond forming, it's always anabolic. And here's an example: amino acids bonding together to start to form a protein. Decomposition reactions, A plus B goes to AB goes to A plus B. So it's just the reverse synthesis reaction. You break bonds, and we call that a catabolic reaction. Here we have decomposition of glycogen into glucose molecules. Exchange reactions, also called displacement reactions, bond both broken and made. So A plus B, it gives this B away and picks up this guy C. So you have AB plus C goes to AC plus B. Okay, and this is showing an exchange reaction. Glucose and adenosine triphosphate. The glucose accepts a phosphate group from the adenosine triphosphate, and so now you have glucose phosphate plus adenosine diphosphate. Okay, chemical reactions either give away energy or accept energy. Now, in chemistry class, we talk about endothermic and exothermic because heat is actually released. But in most biological systems, we don't get a tremendous temperature change, even though things are being broken down, as in catabolic reactions, and energy is released, and as in things being built up, which would be a inorganic reaction. It would be an endothermic reaction if we were talking about a typical chemical reaction. Exergonic, release of energy. This is catabolic reactions. We're breaking down nutrients into their basic component parts. We can absorb them, and then we're digesting them and breaking them down and releasing energy. Inorganic products contain more potential energy than the reactants, And so you're actually building things up, anabolic reactions. This fire-breathing dragon is pretty easy to see. He is releasing energy, but he's also releasing energy as he breaks down to fly down and breathe fire on you. This field of trees, we have an endergonic reaction. Photosynthesis from the energy from the sun comes down into the green grass and causes photosynthesis. So the energy from the sun causes carbon dioxide to form chains. You get sugars and then complex carbohydrates. And even though heat is not, absorbed, you don't get colder, you still have energy stored. So it's endergonic reaction. Then to further the cycle of life, a cow eats the grass, it breaks down those carbon chains, reassembles them into cow carbon chains, and then you eat a hamburger, you break those carbon chains down, and when you breathe out your carbon dioxide, basically you're breathing out the carbon dioxide that came from the cow, that came from the grass, that came from the atmosphere. Okay, chemical reactions may be reversible. You can have A plus B goes to AB, and then it may go down again into A plus B. And chemical equilibrium occurs when you finally get to a place where you get no more reaction either way, either forward or backwards. And many biological reactions are irreversible because of energy requirements, and also products are broken down. Now, this is a person who is about to pass out, and we're just going to assume that this person hyperventilated. Now, we know that carbon dioxide plus water yields carbonic acid. So if you breathe extremely fast and hyperventilate, you lose acid. So you get a respiratory alkalosis. And when you have the respiratory alkalosis, it causes vasoconstriction of the cerebral muscles, and you pass out. If, on the other hand, someone breathes terribly slowly, such as on a surgery table, they will actually build up carbon dioxide, which means they will build up carbonic acid. And they will end up, even though they have adequate oxygen, they can die from a respiratory acidosis. Now, when a person is in a diabetic crisis, they have ketone acids built up because as they break down fat to try to survive, they can't absorb the sugar into their cells. They break down fats, ketone acids are released, and they have a tremendous acidity in their bloodstream. And so they will respond to their metabolic acidosis by breathing more rapidly, and they blow off carbonic acid, and it probably does help them survive until you get them some medical help. Okay, the rate of chemical reactions, and again, these didn't translate very well, is influenced by temperature. The higher the temperature, the higher the rate of the chemical reactions. Now, all these are based on, you have to have a collision to get a chemical reaction. You can't get a reaction if the two chemicals don't touch. So temperature, the hotter it is, the faster the particles move, therefore the faster the reaction rate is. Smaller particle size causes increased rate of reaction. Smaller particles move more rapidly because it doesn't take as much momentum to get them going. Just think about your 2-year-old. Who can run faster, you or him? You yell no, they run in circles, and they're very small and you can't catch them, and they are very likely to have a collision with something you didn't want them to collide with. Now, concentration of the reactant increases the rate of a chemical reaction. Increased concentration. The more particles you have, the more likely they are to react together. Now, a catalyst increases the rate of a chemical reaction without being changed. Enzymes are biological catalysts, and they do this by decreasing the amount of energy it takes to start up a chemical reaction. Okay, we classify compounds into inorganic and organic. Inorganic Inorganic water salts, acids, bases, they don't contain carbon except for carbon monoxide and carbon dioxide are considered to be inorganic because they don't have to be made in an organic system, in a living system. Organic compounds, carbohydrates, fats, proteins, nucleic acids, they all contain carbon, large, and they're covalently bonded. Water makes up 60 to 80% of the living cell, and it's very important in living organisms because of several properties. High heat capacity. It can absorb and release heat with very little temperature change. High heat evaporization. Evaporation requires a lot of heat, so therefore if you sweat and it actually evaporates, then it cools you down. Polar solvent properties. It's called the universal solvent, but it is really not. It only dissolves polar things. As we know, water and oil don't mix because the oil is nonpolar. But it dissolves and dissociates ionic substances. It also forms hydration layers around charged molecules and is the body's major transport medium. It's about 55 to 60% of your blood is pure water. Okay, here we have a chunk of salt put in water, and because we have the negative side and the positive side of the water, you see as you dissolve this, put this salt chunk into the water, the sodium and the chloride dissociate because of the old I'm attracted to your partner thing. This is the negative side of the water is attracted to the positive sodium. This is the positive side of the water attracted to the negative sodium. So the salt crystals will dissolve and spread out and be dissociated. Now, water is part of many reactions, part of hydrolysis and dehydration synthesis. It also is for cushioning protection of the brain. And here we have a fetus growing protected by amniotic fluid. Okay, salt is an ionic compound that can dissociate in water, and it contains cations other than hydrogen and anions other than hydroxide. Ions, or electrolytes, conduct electrical currents in solution, and they play very specialized roles in body functions. Sodium, potassium, calcium, and iron we'll discuss in a lot of different contexts here. Okay, acids and bases, they're both electrolytes. Acids are proton donors because all a hydrogen ion is just a proton, because once you get rid of that electron, which it's going to do, it's just a proton, so hydrogen H plus. Well, we're having oh man, I don't know if this will work or not. Okay, hopefully report any ugly words. Okay, bases are proton acceptors. They take up hydrogen ions from solutions. So sodium hydroxide is a base. The OH accepts the proton, hydrogen, and you get water. Bicarbonate ions and ammonia are important bases. Acid solutions contain hydrogen, and as the acidity increases, as the hydrogen ion concentration increases, the acidity increases. Alkaline contain hydroxide, so the acidity decreases or the OH increases, your alkalinity increases. Now the pH, or acid-base concentration, is a measurement of acid-base concentration. pH is calculated by measuring the hydrogen ion concentration in moles per liter and then calculating the negative logarithm of that answer that you got for your hydrogen ion concentration. Neutral water has a pH of 7. So we have here our a pH chart here. 7 is neutral. It goes all the way from 0 to 14. These are very acidic. These are very alkaline. Blood pH is 7.4, and you don't get a whole lot of variation in that. If you do, you have a physiological problem. Okay, now, since the pH scale is logarithmic, a pH of a solution of 5, a pH of 5 has 10 times more hydrogen than pH of 6. pH changes interfere with cell function and can damage tissue, and a slight change in pH can kill you. It's regulated by the lungs, as we already mentioned. As you breathe out basically carbon dioxide, you're breathing out carbonic acid. By the kidneys, if you need to keep some acid back, it retains the hydrogen ions. If you need to get rid of some, the kidneys dump some. And then buffers are substances that keep the pH from changing too rapidly. The example here, bicarbonate dissociates and becomes a weak base. Organic compounds contain carbon. They're unique to living systems, carbohydrates, lipids, proteins, and nucleic acids. Many are polymers. A monomer is a single unit or building block, and then they're put together into polymers. They're synthesized by dehydration synthesis, broken down by hydrolysis. So here we have monomer 1, monomer 2. You remove water and you have them linked by a covalent bond. Now, if you want to break that link, you add a water in, OH, and H is added, and now they're broken. And here's an example of glucose and fructose. Dehydration synthesis turns it into sucrose, which is common table sugar. Hydrolysis, water's added in, and they're broken down into glucose and fructose. Carbohydrates include starches, sugars, and we have 3 classes: monosaccharides, disaccharides, and polysaccharides. The function for the source of fuel and structural molecules for ribonucleic acid as well as deoxyribonucleic acid. Now, these are some monomers, monosaccharides: glucose, fructose, galactose, deoxyribose, and ribose. These are some disaccharides. Di is for 2: sucrose, maltose, and lactose. Then we have polysaccharides. Glycogen is an example. Lipids, essentially your fats and fat-type substances. They contain carbon, hydrogen, and oxygen, and sometimes phosphorus. They do not dissolve in water because they are nonpolar, and we have several types we'll talk about. Triglycerides are neutral fats. They're solid fats or liquid oils. Oils come from plant sources, fats come from animal sources. And they're composed of 3 fatty acids bonded to a glycerol molecule. The main function is for energy storage, insulation, and protection. And here you have a glycerol, 3 fatty acids. They're bound together and you get 3 molecules of water. Okay, saturated versus unsaturated fats. I have a picture here of a saturated fat. Every bond is a single bond between the carbons, and you have all the hydrogens you can get. This is unsaturated. Now, the more double bonds you get, the more likely you are to have a liquid at room temperature. Now, why do we care about fat in our saturated fats in our diet, and that's because increased chance of heart disease and other vascular issues are associated with saturated fats in our diet. And I kind of like this cartoon. He says, instead of jogging, can you just set my pacemaker to beat faster for 30 minutes a day while I watch TV? Sounds nice. Phospholipids, these are modified fats, modified triglycerides. It's a glycerol, 2 fatty acids, and then a phosphorus-containing group. It's a very important component in the cell membrane. It has a head and a tail region. The head region is polar, so it dissolves in water. And the tail region is, no, I told you backwards, I'm sorry. No, I didn't. The fatty acid side is nonpolar. So you have the polar head here, the nonpolar tail. So this side is water soluble, this side is non-water soluble. Steroid is cholesterol, vitamin D, steroid hormones such as sex hormones, and salts are all formed from steroids. This is an example of a simple steroid ring. And in case you don't know, each of these corners represents a carbon molecule, each corner with nothing written on it. Because steroids, there are many different ones. Prostaglandin is one we talk about because it is involved in inflammation. It is an instigator of inflammation. Other lipids in the body, fat-soluble vitamins, vitamins A, vitamins E, vitamins K, and vitamin D, which is not listed here, D3. Also lipoproteins that transport fats in the blood. Proteins are polymers joined by peptide bonds, polymers of amino acids, and there are 20 different amino acids. Carbon, hydrogen, oxygen, nitrogen, sometimes sulfur, and phosphorus. And these are showing some general amino acids. And then you see they've had dehydration synthesis to put them together. They can be broken down by hydrolysis. Now, this can only take place in a living organism. You can't make a peptide bond, only, in a chemistry lab. Only organism can do so. Now, here we have a primary structure of a protein, a polypeptide chain. It would probably be a lot longer than that. And then because of hydrogen bonds, attracted to that, this is attracted to that, they start to change shapes and curl or maybe form sheets. And then as they continue to form, this is a tertiary structure. You have very many bends and turns, and this is a quaternary structure. Many bends and turns, and it's a very complex structure. Now we have 2 types of proteins, fibrous and globular proteins. Fibrous are also structural proteins. They're strand-like, water insoluble, very stable. Keratin, like in your hair and nails, elastin, collagen, contractile fibers, actin and myosin. Globular proteins or functional proteins, these are sphere-shaped, they are water soluble, and these are things like antibodies, hormones, molecular chaperones, and enzymes. Now, protein can become denatured. If it's just a little bit of change, it may be reversible, but extreme change, such as extreme heat on a protein, it becomes irreversible. You can't uncook that egg. Now, if you feed that egg to a chicken and he lays it, it's still an egg. It's not the same egg. This has been irreversibly damaged. So if you leave your kid in a car and they get sitting in 120 degrees, their brain cells and their body cells will become irreversibly damaged. Molecular chaperones are chemicals that help proteins bend into the right spaces to be formed. They fold properly, and they help trigger the immune response, and they respond to stressful stimuli. Enzymes are biological catalysts, and they the activation energy of a chemical reaction, and they increase the speed of a reaction so that you end up getting millions of reactions per minute. So this is if you did not have an enzyme, it takes a lot of energy. If you do have an enzyme, it takes much less energy. Now, they're often named for the structure they catalyze. They may end in the term ACE, like peroxidase breaks down hydrogen peroxide. Now, some enzymes, the functional enzyme or holoenzyme, has the apoenzyme, which is your protein, and then a cofactor, which may be a metal, or a coenzyme, which is going to be a vitamin. This is showing you a little about an enzyme. You have two substrates here. They, by using like a lock and key, they fit together and you get hydrolysis, and now you have your peptide bond. And enzymes work strictly in a lock and key fashion. The enzyme is used over and over. Nucleic acids, DNA and RNA, these are the largest molecules in the body. They contain carbon, oxygen, hydrogen, nitrogen, and phosphorus. The building block is a nucleotide composed of a nitrogen-containing base, a pentose sugar, and a phosphate group. Deoxyribonucleic acid, the four bases are adenine, A, guanine, G, cytosine, C, thymine, T. They are double-stranded helixes, and these contain the code for protein synthesis. Now, when a cell is going to divide, they have to replicate, or basically make a copy of each side, so they'll unwind and copy each side, and then you have two sets of DNA. Here is your double helix, and it shows it's just starting to unwind here. Ribonucleic acid is a single-stranded molecule, and it takes the message from the DNA out into the ribosome so you can make protein. Adenine A, guanine G, cytosine C, and instead of the T, you have uracil U. And there are three varieties of RNA that are involved in making protein: messenger RNA, transfer RNA, and ribosomal RNA. Now, adenosine triphosphate, ATP, it is essentially the charged battery for your cell. When you break down glucose, the whole point is to make ATP out of adenosine diphosphate. This is showing charging up a cell phone. Well, when you charge up, when you take adenosine diphosphate, you store energy in that extra phosphorus bond, and then it can be released to do things that the body needs to do, such as live, breathe, exist, all things that require energy.